Our next speaker is not here, but he is here. It's Luis Diaz, who's the new selection for, or candidate, not beyond candidate now. He's the new rootstock breeder at UC Davis, so he's taking my spot. He'd be not on my desk, but he'll be somewhere in the building. And right now, he's somewhere in Mexico. So Luis finished his PhD in Wisconsin and was working on a postdoc in northern Mexico, and he'll give you a little bit of that information and what's coming next for him as well. So, Luis, take it away.
Hello everyone, I'm very happy to be here with you today, at least virtually. I hope to see you soon, maybe just in a couple of months. Just wanted to start saying that I'm very happy and very excited to join the Viticulture and Enology department at UC Davis. My name is Luis Diaz Garcia. I am from Mexico. Some of you know me a little bit. I have been talking with some people, some faculty, students in the last few months. I got my bachelor's degree in agronomy from Universidad Autónoma Chapingo, which is like 30 minutes from Mexico City. Then I got my master's degree in plant biotechnology, also here in Mexico. And then I moved to the US to get my PhD. I was in the University of Wisconsin-Madison, where I work in cranberry genetics and genomics. Grapes are arguably the most important fruit crop in the world. There is no doubt of its importance in terms of economic revenue, jobs, science, and history in general. So because all of these, there is a tremendous amount of resources that we as researchers and breeders and extensionists can use to improve this crop, to make new varieties, to come up with strategies for manage our vineyards in a better way. This was not the case for cranberries, and this is not the case for many of the crops we grow in developing countries like Mexico. When I joined the Cranberry Genetics and Genomics Lab, this was in 2014, the molecular resources to accelerate breeding in cranberry were very limited. On that time, the only tools or knowledge or resources that we had included just a draft linkage map with just a few SSR markers and less than a dozen marker trait associations or QTLs. For the ones that don't know what this means, a linkage map is some sort of map that can tell us where are the important regions in the genome that regulate phenotypic variation. If we are talking about cranberries, this variation can be anthocyanin pigmentation or fruit size or fruit yield. If we are talking about grapes, this variation includes resistance to certain diseases, skin color, fruit, berry size, cluster architecture, among others. So having a linkage map is like the first step to get a more efficient and a more accelerated breeding program. Some people call this marker-assisted breeding. I spent the first 2 years of my PhD working on cranberry genetics and genomics and developing these molecular tools that could be useful for accelerated breeding. Then towards the end of my PhD, I focused more in phenomics, which is a new research area that is focused on the developing and implementation of high throughput methodologies to gather lots of phenotypic data. All my work in cranberries was always aligned with the needs and priorities of the industry, but this is changing very quickly. It's a very dynamic thing. In the past, researchers and breeders were more focused on increasing anthocyanin pigmentation or fruit size or fruit yield. But as I told you, this is changing and this is very dynamic. So now, because of the profits from selling dried cranberries, researchers and breeders and scientists in different institutions are focusing more on things that are related with the firmness of the fruit. After finishing my PhD in 2018, I moved to Mexico. I got a position at INIFAP, which is like the USDA. It's a government agency for research in horticulture, livestock, and forestry. I'm currently located in Aguascalientes, which is like 5 hours north from Mexico City. In the past, Aguascalientes was the most important region in the country for the production of table grapes and wine grapes. There were lots of vineyards and winemaking companies. And this is a picture of my dad harvesting grapes in the late 70s. And also you can see here the state logo. I think it's also known as the state coat of arms. There are different elements here. Each element has a meaning, but one of the elements is a grape cluster, and all of this is because of the importance of this crop for the state. Sadly, this has been changing in the last few years. Now we don't have that many grapes. Most of the agriculture in the state is dedicated to forages for milk production. Towards the west part of the state, we have a region we call Los Cañones, or the Canyons. It's a region characterized by a more subtropical climate. We have higher temperatures and more rain, and it's a region that produces lots of guava. Maybe you know this crop already. Guava, it's a fruit crop. It's very tasty, has a lot of vitamin C and other nutrients. And similar to cranberry, the molecular resources for guava are very limited. And this was not new to me, so I decided to work on guava here at INIFAB. So I'm very confident that my work in guava will set up the foundation for future researchers and breeders to improve this fruit crop. So these are some of the things that I have been doing in the last few years here in Mexico and in Wisconsin, working in different fruit crops. Now I would like to take some minutes to talk about the perspectives and viticulture and grape breeding and some of the achievements Andy Walker have done in the last few decades. Andy Walker and the ones that were before him built a world-recognized breeding program. His achievements go beyond the releasing of his URN roostocks or the PD-resistant cultivars that he just released. And the germoplasm collection is enormous. It's basically a treasure and a reservoir of alleles and phenotypic variation that is going to be the foundation for the next generation of cultivars. For example, Andy's collection of Vitis arizonica and other related hybrids contain the genes to achieve long-term resistance to Pierce disease. Also, his large collection of other species from the southwest of the US, for example, contain the alleles required to gain resistance to other diseases, pathogen, or stresses in the environment. So part of my job at UC Davis would be to assess the best combination of alleles in order to obtain better and more adaptive grape cultivars. One way to accelerate the process of selection and breeding, and particularly in fruit crops like grapes, is through the use of molecular markers. And this is not new to grapes. Both the industry and public universities and other institutions have been using molecular information linked to several traits to predict phenotypes like flower sex or muscat aroma, or resistance to certain diseases like Pierce disease, quality traits, and a few others. And that has been helping breeders and researchers to accelerate the breeding process by saving time and space in the field. So in this case, selection is done based in just a few markers, like a dozen, I think, particularly those traits that are linked to major traits. So the other way to employ market information, but this time using data from the whole genome, is through genomic selection or genomic prediction. Genomic selection is a trend in breeding in many crops. There is certain debate if this is useful for grapes or not. So from the breeding perspective, grapes are very peculiar. And this is maybe why genomic selection might not be useful. Just for comparison, other fruit crops like strawberries or apples are more dynamic. I think cereals are also the case. So by dynamic, I mean that the rate of development and adoption of new cultivars is much higher than in other crops like grapes in this case. So this is just an example of the strawberry breeding history from a study published recently by the strawberry breeding team at UC Davis. Every point here represents a cultivar starting more than 300 years ago. And the lines are connecting two cultivars, and this is the relationship, parent-offspring, between these two materials. You can clearly see a very dynamic history of replacement, which means that certain materials appear over time, certain materials disappear, certain materials are more commonly used as parents because of the desired characteristics these materials have. And in grapes, the story is quite different. We have materials, what we call the traditional cultivars, that were generated hundreds of years ago and dominate the market right now. And these materials, some of them have an origin, some of these materials are the parents of hundreds or even thousands of materials that we can find all over the world. So this is like a more static version. Cultivars don't change as quickly as in other crops, like the example of strawberries. So we could say then that the demand for new cultivars might be very low. So tools like genomic selection, which aims to accelerate the breeding process, might not be very useful. But we know that there is an increasing interest and increasing trend to adopt new cultivars, particularly in progression lines that have just a small portion of wild genomes, the regions that are linked to desired characteristics like resistance to certain diseases, but most of the genome is basically beneficial. So maybe genomic selection is going to be an important tool in those cases. Also, rooster breeding might be more dynamic and open to new materials. So I think these two could have a chance and might be a good fit for breeding roosters. So I would like to take some of the data we have already. There is plenty of genetic and phenotypic information, and to conduct a comprehensive study to know about the feasibility of genomic selection for breeding, particularly roosters. My PhD advisor used to tell me that plant breeding was about numbers. The more plants you evaluate or the more crosses you make, the more chances you are going to have of getting new cultivars. And then I know that this might not be true in some cases. It's very specific to certain circumstances, to the germplasm we use or the species we're working with. But I will argue that in general terms, plant breeding, it's a game about numbers. So how do we increase the plants we evaluate in a breeding program? So part of my plans at UC Davis involve the implementation of high throughput approaches like rovers and drones to assess the physiological response of different materials to different stresses in the environment, or computer vision to quantify and model root architecture, and maybe combine all of these things to come up with new traits that can tell us about the potential of certain germplasm for the production of new cultivars. I think that the challenges we are going to see soon, or probably we are seeing now, are going to require us to come up with better and more multidisciplinary ideas to solve the problems of the viticulture industry in California, as well as in other parts of the country and the world. Water is going to be an issue. Labor availability is going to be an issue. So we'll have to come up with strategies to produce varieties that can deal with these deficits on water, but also with better vineyard management practices that can allow us to mechanize certain activities in the vineyard. So tools like molecular assisted breeding, genomic selection, or computer vision are going to be very useful to make this process more efficient. So just to wrap this up, I just wanted to say that I'm very happy to be here with you today. I'm really excited to join the Viticulture and Enology department. It's such a great faculty and students and staff and facilities and history in general, more than 100 years. I'm particularly looking forward to continue on this legacy of viticulture and grape breeding, not only in California, but in the country and around the world. I'm particularly looking forward to maintain and strengthen the collaboration relationships Andy built with the industry for more than 30 years. So again, thank you very much for having me here, and hope to see you all very soon. Thank you.
Thank you, Luis. He's somewhere out there. Lunchtime? No, one more?
They're going to try and bring him. There he is.
Ah,
Hello, everyone. Hello, Andy. I'm here.
good, good, good.
Anybody has questions?
Well, I don't know. Can you hear me?
Any questions for Luis?
Well, Andy, just before that, I just want to add a couple of things. Thank you very much for having me here. I was fascinated listening all the stories about you, by all the people that was talking this morning. I'm very happy to join the department. Hopefully we can have you in the team and help us, continuing all the work that you have been doing in the last few decades. It was very, the story about the sticky notes was very funny to me. And I will probably be teaching some of the classes you were teaching in the last years. So I will be doing that, and I have to learn, and it's not going to be easy. Probably I will be the one putting sticky notes in the leaves. So I will be, a better professor for the new students coming to the department. So again, thank you very much for having me here.
You're very welcome. The sticky notes legend has gone on for quite a time. If I was smart, I would have done something different. So I took those tags off, put them in a big pile, and tried to figure out whose handwriting it was. There were about 9 candidates. And during that exam, when I decided, well, we just won't put the sticky notes back again, I just watched the response. And there were more than 9 people were searching for sticky notes. So it was very amusing. I should have just mixed them up, right? I should have had Cabernet. We've been very clear that way. It's a good trick, though, Luis.
Okay, yeah, I'll be very happy to bring that legend back again.
Any questions, Louise? They're being nice to you.
They're tired.
Yeah, I was very happy that my talk was exactly before the lunch break, so not many questions.
Okay.
Well, thank you.